talk useful. As Hernan was mentioning, I'm a physics professor at the University of Buenos Aires, and today I'm going to talk to you about this CASPAM, which is a triple modality biosensor that we have developed for multiplex imaging of caspase network activity. In our group, we aim to walk between three things. We try to understand the three-dimensional organization of living matter, and for this, many times we find that we don't have the methods that we need to query those systems. So we develop optical quantifications of these molecular processes, and then we have to deal with the intrinsic biological variance which we found when we do these experiments, and therefore we try to develop a statistical method for this system biology.
Hernan? How about the rest? How about the rest? The other people is connected? Okay, Florencia say yes.
And proliferation is the only truly serious biological problem. As some of you already know for sure, apoptosis is a form of programmed cell death. Unlike necrosis, in which cells swells and after rupturing the plasma. Yes?
Hernan, can I interrupt you because we lost you for a minute or so.
Okay.
When you were, yes, I think it will be nice if you can come back because we.
Sure, no problem.
Yes,
where we lost?
We lost you here basically.
Okay.
And it looked like you have in the bars it's like in yellow. Maybe.
Okay. Let me do the following. I will unplug my Wi-Fi and then just be sure that I'm wired. Okay, that the wire connection is effective.
Great.
But give me 1 second for that. Okay, I'm connected now via cable. Do you hear me? Yeah, perfect.
Yes,
okay.
Very well. Sorry for interrupting you.
No, please. So I was saying that in my group, just let me recap from here. In my group, we try to understand the three-dimensional organization of living matter. And for this purpose, many times we find out that we don't have the methods to achieve this. And therefore, we do the development of optical quantifications, methods for molecular processes, and also we develop statistical methods to deal with the intrinsic variance of biological systems. Let me make a small detour, and I would say that in this book, which is The Myth of Sisyphus, Camus argues that the question of suicide and the meaning of life is the only truly serious question in philosophical problem. And I would argue that if Camus was a biologist, then he would say that apoptosis and proliferation is the only truly serious biological problem to study. Of course, a lot of colleagues will not agree, and I'm sure there's a lot of other things interesting to study, but I was really motivated to study apoptosis because, as many of you know, this is a form of programmed cell death, right? And unlike necrosis, in which the cell swells and after rupturing the plasma membrane, the cellular context spills over the organisms, causing inflammation, during apoptosis, the cell breaks into several bodies, which are then phagocytosed. This process of dying is essential for living, and it happens during development for removal of tissue and organ sculpting, but also in adults from apoptosis. And it has to be really tightly controlled. And how this happens, it was a large mystery to me. And also I found out that it was a fantastic process to develop methods to study biological systems. And let me tell you why. Apoptosis is controlled by a biochemical protein network, which is the one that you see on the screen. It basically transduces information from two different sources. On one side, you have the intrinsic pathway, which integrates information about the cellular state, such as DNA damage and stress. And in contrast, the extrinsic pathway is activated by extracellular ligands binding to cell surface receptors. Then both pathways converge downstream into what is called the effector pathway. And interestingly, these pathways are highly conserved among different organisms because, of course, apoptosis is a very important process. And also, an essential protein in this network are caspases, which are a specific type of proteases. And proteases are just proteins that cleave other proteins. And therefore, it's rather easy to build a caspase activity sensor by a cleavable substrate. And this indeed what it was done, at least in 2008 and maybe before also, in which they build up two fluorophores, let's say a green emitting fluorophore and a red emitting fluorophore, which are a good thread pair. And you know this already, thanks to the course that the lectures that you have been having. And then with the protease will cleave the linker that is connecting these two fluorophores, and they will drift apart. Therefore, you will go from a moment in which there is thread between the red and the green protein, and then to a moment in which there is no thread after the protease is active. Therefore, a protease activity sensor, it's something straightforward to do, unlike other sensors, which is much more complex. Of course, this sensor is not perfect. For example, you cannot measure deactivation, so you can only cleave and not uncleave, which will be nice. It's not reversible, but it's a good start to do many things. And the problem with these sensors is that, of course, allows you to measure two colors. Maybe you can squeeze another two colors, but you know the visible spectrum, it's kind of narrow, and the fluorophores, the fluorescent proteins, are kind of broad in the spectrum. Therefore, there's a limited number of fluorophores that you can put into the spectra and quantitatively and rigorously image and quantify the information in the single pixel level. Therefore, another way to do something like this and to make use of this thread is by using polarization anisotropy, which also can be used to measure sensor state. Briefly, what you do is you shine polarized light over an ensemble of sensors, fluorophores, which can be in any orientation. And so those which are in the right orientation, which is in which the polarized light has the same direction as the excitation dipole, will be excited and therefore will emit, and therefore the polarization will be have a high degree of anisotropy. There will be more light in one polarization than in the other. But there are processes that may depolarize this light. One is rotation, so the molecule can be excited and then start to rotate, and therefore the emission will be decoupled from the excitation polarization. But another process that also depolarizes emission is FRET. And therefore, just like in here, let's say, for example, the sensitized emission or the donor quench emission can be, or the lifetime can be used to know if the linker is bound or unbound. Here, the polarization anisotropy can be used to determine the same thing. For example, when you excite with polarized light an ensemble of sensors which are all linked to each other, then the emission will be more or less the same in both directions. It's actually not true because there's some intrinsic anisotropy, but there will be low anisotropy. And then when you start cleaving, so the CASPAM is activated, which is symbolized here by this scissor, then what will happen is that the anisotropy will be increased a little bit. So the polarization, the intensity of the polarization in one direction and in other will be more different. And finally, when all the sensors are cleaved, then the emission will be highly anisotropic, and because all the fluorophores will be by themselves. Importantly, we are using here fluorescent proteins, which are rather slow to diffuse, so you can mostly consider it as static dipoles in space. Okay, we can go over the details of this in the future. So already some time ago, what we did is start using these sensors to monitor the process of dying. So when the cells start to die, what you see is that the cell starts to swell, and also there's bleeding. And you see in the lower panel, the anisotropy value goes from a low value, which let's say 0.20 something, to a higher value, 0.30 something. And so basically, by monitoring the shape, we have a phenotypic view of what the apoptotic process is going on. But by looking at the polarization anisotropy, we can have a molecular readout of when the decision is being taken. And so indeed, what you can do is have add a stimulus, for example, staurosporine or methylene blue in solution, or any other reagent that causes stress to a cell or a death ligand, And then what will happen is that the caspase will be activated. And by monitoring the anisotropy, you can see when the cell has decided to die. And you see, again, low anisotropy, the fluorophores are together. High anisotropy, the fluorophores are apart from each other. And here we have so the trick is to have the right linker. So here is DEVD, which is a linker recognized by caspase 3, but then there are other linkers recognized by other caspases, so other points in this network. So 2 important features. First, this is a ratiometric technique, because to calculate the polarization anisotropy, you have to do a ratio of intensity quantities. So it is very important that you do 2 things properly. One is that you do a correct background subtraction, and then that you have a good signal to noise ratio. So let me tell you how we deal with those 2 things. First, we achieve proper denoising by a technique that we developed that is using wavelets. So basically, many times when you denoise, what you do is you average out features, but that is not what you want to do, because the cell might have some structures which are very well defined in space and some others which are more spun out. So wavelets is a way of denoising that allows you to conserve structures which are localized in space by averaging pixels in a way which is a varied size, varied grain size. So some areas you average a lot, and some areas you average very little. The question is, how do you decide which pixels to average out? And for this, we do a statistical test. Many times what people do is they compare the intensity. But for example, for this paper that you see here, we did it for film, so I don't have to explain you this, but we show that by using higher harmonics to calculate the covariance of these clouds in the phasor plots, that is a proper way to do a statistical test to decide which pixels should be averaged out and which pixels should be kept apart. And with this, we have a very large reduction. For example, this is a simulation in which we have a ground truth. And here you see the undenoised phasor plot. By the way, Bob Clegg did something like this in the past. So this is using Bob Clegg's method. This is a normalized version of Bob Clegg's method that we derive as well. And this is using this POFLIM algorithm, which take a look at the image here in the inset, but also on the clouds. Look how much better these clouds look like because they have been denoised while keeping the spatial structure. And we have shown this, of course, for real data, and it works really nicely. We do this method also for fluorescent anisotropy images. And the second part is that we can use the lack of spatial correlation in the background to obtain an accurate estimation of the background distribution. Here, what you see is an image on the real image, and then in orange, a small region of interest in which there are no cells. And so you see the cumulative distribution function, or the PDF here below in orange, which corresponds to that. So if you use 2 common methods like Otsu thresholding or rolling ball to find the background, what we find is that these methods, they are highly biased. And this depends on the number of cells that you have, how filled the image is, and etc. By using our method, which we have called SMO, we basically do, again, a statistical test to decide which pixels have little correlation with the nervures, and then take a sample of those and get a proper estimation of the background distribution. And these 2 things give us much better results to obtain in our ratiometric imaging. By the way, so we have submitted this paper now, and you can find it in bioRxiv, but there's also plugins for Napari, Fiji, and Cell Profiler if you want to test it out and give us some feedback, because this is, of course, very new. Okay, so with these 2 ideas, then we can properly go to quantify the fluorescence anisotropy. And then you can think that, the protease is the scissor that cuts the sensor. So we don't really care about the sensor. We care about the scissor activity. So basically, by doing kind of a derivative, you can find the activity in arbitrary units of the protease, which is what you want to do. And this was described in this paper in Grecco's biology. And we also described there that you can now make good use of why we shifted from, let's say, heterothread to, let's call it homothread or pseudo-homothread or pseudo-heterothread, and I will tell you why. So basically, now we can put 3 sensors, each of them spanning a narrower window than a typical heterothread sensor. So we have a yellow sensor composed by 2 M-citrine fluorophores, that has a ligand for one caspase, a red sensor which contains mcherry and mkate for another caspase, and a blue sensor which contains BFP and cerulean for a different caspase. So it's not really heterothread, it's not really homothread completely, it's a mixture of things. But this gives us the best signal to noise ratio in the polarization anisotropy. And then for each cell, we can measure the anisotropy as a function of time. And what you see is that at a certain point, the different sensors rise and the polarization changes dramatically. And below, you see the activity of the sensor. So interestingly, we did this over 15 hours for a lot of cells. And some cells start to die at the very beginning, some cells start to die at the very end. But because we measure the three sensors, we can now calculate the difference in activation of one sensor to the next. And we can do this type of histograms in which we measure, for example, the difference in time between caspase 3 activated and caspase 8 activation in the y-axis, and caspase 3 activation and caspase 9 activation in the x-axis. And we can, and these histograms provide an overview of how the cell is reacting to a specific stimulus. And by the way, I don't want to go into detail for time constraints, but basically this information is not just photophysical information, but this is biological information, which you can model and you can compare with pre-existing models. So this in blue is a model that was published before us, which was fitting, let's say, one axis of our experiment, but not the other one. And then we are able to modify this model to create an improved version of this extrinsic apoptotic reaction model. But this technique has two problems, at least two problems. One is that the timing of the sensor depends on the sensor concentration. This makes a lot of sense, and it's true for every sensor. If a sensor is a substrate of the caspase that you want to measure, if you put more substrate, the substrate will react slower. The second problem is that not all cells have a useful fluorescence channels, fluorescence level, sorry. For example, it might be that some cells are very bright and also some are very dim, but this dim or bright will be different in each cell. So we wanted to have is a way to control the intensity of the expression. And so for this, we created this plasmid, in which we use 2A peptide cis-acting hydrolase elements to co-express from a single open reading frame. So basically, CASPAM is these three sensors that I told you before in a single plasmid. So then they are expressed, so they internalize into a sensor, they are expressed, and they generate three different proteins, but which are basically in equimolar concentration. We check this with different methods. For example, here on the right, you see flow cytometry measurements, and you see that with CASPAM below, you have a very nice correlation of the two channels, and actually I can show you also for the three channels, it's in the paper. But if you have the separate sensors, you have some cells which have a lot of one sensor and nothing of the other, and vice versa. And this produces problems when measuring the timings. And so with this sensor, we went to our second goal, which is why don't we use the same process, not to study the extrinsic pathway, but also to study the intrinsic pathway. So we have a batch of cells, some we stimulate with extrinsic stimulus, some with intrinsic stimulus, and then we measure how the network responds to those stimulus. Because interestingly, it's the same network, but in the literature, it's full with papers which have a model to explain the response to extrinsic stimulus and another model to explain the response to intrinsic stimulus. But it's the same network, so it should have a single model to explain both responses. And so these are the results in these two-dimensional diagrams showing specifically that. We did this for several hundred cells, and we measure at what time the different caspase was activated and the difference between those. And we were able to find out by modeling this network that there is an extra feedback that was previously unknown between caspase 3 and caspase 9, which is needed for activation. And to me, this makes complete sense, because if you have a process which is involved to kill a cell, you want that this process is definitively. So once that the cell has committed to die, it should die. It should not stay in a zombie state. Therefore, this extra feedback reinforces the decision of dying. And our data points into that direction also, as you can think of how the process works. So to sum up, basically CASPAM this sensor that we developed allows to simultaneously measure the effector, the extrinsic, and the intrinsic caspase activity at the single cell level. It's very important that we transform these photophysical readouts into biological observables, because those biological observables, and I think this is key to the course, and you have been seeing this in all lectures, are the ones that can be compared to models. And in particular, using these models, we found that feedback from caspase 3 to caspase 9 is required. So with this, I thank you for your attention, and I open to any questions that you might have.